We're going to get started looking at the basic structure of DNA and RNA in our notes today. We're going to take a look at what this foundation really is in between DNA and RNA, and we're going to start off with looking at some of the basic notes and why do we need to know about it. DNA carries instructions for all life's purposes, and one of the things that we're going to see is we're going to draw it out and see what it actually looks like when we have it. Now, we've been going through a whole bunch of different notes on the structure of DNA, and just kind of quickly draw out a couple lines of it. We are going to see inside there structural components that are going to make it up. These little red dots, dashes, are going to be our nitrogen bases, that are going to be our rungs of our ladder, and the two lines on the sides are alternating sugar and phosphate backbones. Now, the second bullet point there is asking you, what is a gene? Now, we just got done talking about genes and the fact of Mendelian genetics, all about these guys are the alleles, because they're coding for particular traits. Now, a gene is in this section of DNA. So that's what a gene is, a section of DNA that codes for a protein, and that code is a trait, and the trait is the form of a trait, is that allele. So those two things are what really tying together from our last chapter. We talked about which traits are you inheriting, or which alleles are you getting, are based upon the genes that are coded from within inside your DNA. What is the function of it? Serves as, again, a code for the protein. And why do we need to know about it? As we continue to learn more and more about diseases, everything from things like cancer to things like Tay-Sachs or PKU, the more we know about the genetics or the genome, the more and more we can improve medicine. In fact, now you can even you can YouTube a video on preventative medicine. The Mayo Clinic is looking at ways in which we can now do preventative care versus reactive. You'd rather go to the hospital before you get sick and prevent it from ever happening than it would be to whenever you're sick or you know that you're going to be having cancer. If we can treat it before it even happens, better chances of survival. So the more and more we know, the better chances you can live. Go on to what this next step is. It's called the central dogma of DNA. This picture doesn't do a very good job of what it is, but the main idea behind DNA is going from our two-stranded DNA. I'm just going to draw it on its side. So our two-stranded structure of DNA, and how I can convert DNA into an RNA message. And so that's one-stranded. So from two strands to one strand, and then RNA message can code into linear amino acids together, which we call a protein. Now, sections of DNA code into RNA, which codes into a protein, which we call those sections again, those sections in there, a gene. So this main idea, how DNA eventually codes for a protein, is known as the central dogma. Now, disregard some of the pictures over on the right-hand side. They're not working as out as well as they should be. But what is DNA? DNA, the term for DNA actually is called deoxyribonucleic acid. Deoxyribonucleic acid, because when I look at my structure, I am going to have a 5-carbon sugar molecule that's known as deoxyribose. Ribose is our sugar and is without an oxygen. In fact, if I'm looking at this part right here, all of these sugars look the same. This has got an OH group on it, and so does this one. But when that OH group, when this guy right here is absent, we call it deoxy. If it is present, we call it just ribose. We'll talk about in a second RNA. So when it is not there, we call it deoxyribose. When it is there, we call it ribose. Okay. Look a little bit more at the structures of the chromosome. A chromosome structure, we went through all that. We talked about how we have diploid sets in each of ours. We have one from our mom, one from our dad, so we have our 23 pairs of them. Kind of zoom into that again. We have 23 pairs. Replication is the S part of interphase. So as I talked to you guys way back about the G1 S, and G2, all of interphase, where the cell is going to grow up, do its daily functions. But now we just always said DNA gets copied. We're going to learn, in fact, how this copying, this replication actually takes place, and this is all about organelles being copied. So we're going to spend a lot of time in here talking about DNA replication. Now, let's get a little bit just the basis of DNA before you get into DNA replication, and we'll take those notes later on this week. But what we need to know is the structural components of DNA. Do not worry about the purines and pyrimidines, those terms. You will want to know the four names of the nitrogen bases. The four names of the nitrogen bases are adenine, cytosine, guanine, and thymine. And what you would also want to know is that there is a guy named Erwin Chargaff that had a rule. He had found out that there were the same amount of what we call purines as pyrimidines, these two guys here, but what he found out is that he came up with the rule that A's pair up with T's and C's pair up with G's. Now, the pairing up, the point in here, the bonds between those are known as the hydrogen bond. And in fact, between an A and a T, there are two, and between a C and a G, there are three. So 2 hydrogen bonds between an A and a T, and 3 between a C and a G. So whenever we see a DNA strand, so if I'm looking at my DNA strand here, and if I was to zoom in on this, I'd see my strands here. If this is an A, it should have a T next to it. If this is a C, it would have a G next to it. Okay? Now, the basic components of this, you guys all had in biology A when we talk about the biochemistry structure of it. The three parts to the basic structure is called a nucleotide, and it contains a phosphate, sugar, and a nitrogen base. The phosphate, sugar, and nitrogen base, the phosphate group is there, the circle. The sugar is our 5-carbon sugar, and that can either be ribose or deoxyribose, if again the oxygen is not down here. And then we are going to have the nitrogen base, which is going to be the A, T, C, or G. Now, in one of the packets, you guys are actually going to draw out your own structure of DNA. Now, what I'm going to kind of draw out real quick is showing just the phosphate sugar backbone. So this is phosphate, sugar, phosphate, sugar. So on and so forth. Okay, so what I have here is the phosphate groups alternating on the rungs of the ladder and the sugars in between. Now, connecting to the sugars are going to be the nitrogen bases. Now, if this was an A, I have to pair up with a T. This is a G, I pair up with a C. That's a C, I pair up with a G, and if this is a T, I pair up with an A. Now, what I would do then, you can flip this over, and so what I am going to see here is my structural components of, again, the phosphate, sugar, nitrogen base. So on and so forth. Okay, so I'm going to have the alternating sugar, phosphate backbones back and forth on either side. So again, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And there actually should be another phosphate down here. Now, we're going to talk about the structure of this here in a second. But one more thing that's going to occur is when I zoom in on this, here's my structure of it. I want you to take a quick peek at, there's a phosphate group there and a phosphate group there. Missing one here, missing one down there. What that is going to entail here in a second is we're going to talk about the carbon structure. Okay, here is our 5-carbon ring of our deoxyribose and our ribose. And when I count the number of carbons, this is carbon number 1 at that spot. At this spot is carbon 2, carbon 3, carbon 4, and there's actually a bond to a fifth carbon here before the phosphate group. So if I look at my carbons, I call this carbon 1, 2, 3, 4, and 5. And so I'm going to build a brand new nucleotide from, if I want to add a brand new nucleotide on, I'm going to make going from 5 prime to our 3 prime, or our fifth carbon to our third carbon. So if I look at that, how I add those on, if I have a nucleotide, which is my 3 parts, and I want to add a brand new nucleotide to it, so I can take this guy, and I have another nucleotide here. I need to add this to it. I need to add it to the open end. So another 5 prime, and I put a bond between here. Okay? I only add it to the new end. So a new DNA strand is only going to be made 5 to 3, and the prime refers to the carbon. 5 prime to 3 prime. So you're going to have to get that in your head. We're going to go over and over and over again. So again, if I have another nucleotide, here's a structure. I need to convert that. And again, without having to move it over, just do another one. Here's another nucleotide. Put it here, put it underneath, and again, you're going to have a bond between those. So it's just showing you bonds that are between those. Okay? Now, if you take a look at how that was, here again, we're looking at our structures. We have would be a phosphate group here, the 5' end, a phosphate group down here at a 5' end, and it alternates back and forth. Phosphate sugar, phosphate sugar, phosphate sugar. That D is a sugar, which is referring to deoxyribose. So we have our nitrogen bases, which are A, T, C, and G. We have our deoxyribose, a phosphate group, and collectively with all 3 parts, we call that a nucleotide. Double-stranded, two-stranded, and the scientific term that we're using is a double helix. The common term is a twisted ladder. So the difference between scientific and common. Okay, we already mentioned the hydrogen bonds. Between an A and a T, you find 2, and between a C and a G, you find 3. Erwin Chargaff had talked about that. Structure of DNA, base pairing. The one thing that's kind of nice about this is that when I give you an unknown strand, so if I give you a strand, let's just do black here. And I'll say this is my 5' end, 3' end. If I just give you some letters in here, if this is A, T, T, A, C, C, G, you should be able to tell me its components. You should be able to say it's a T, it's an A, it's an A, T, G, G, C. Okay. One of the common rules that we're also going to get into is the rule about anti-parallel, the structure of it. So again, this is where this 5 and 3 comes into play. 5 is always going to refer to our phosphate group. Notice the phosphate group here, phosphate group there, not one down here, not one up there. Those are the open ends that we can add our new nucleotides. So when a DNA strand is made, it's going to be made in an anti-parallel opposite direction, but in a 5 to 3 prime. Important people to remember, we already mentioned Erwin Chargaff. He identified A's to T's and C's to G's. Rosalind Franklin was one of the female scientists that allowed for us for the discovery of DNA. She did what's called X-ray diffraction or X-ray crystallography. She took X-rays of DNA, frozen DNA. Unfortunately, back then, she didn't really realize that X-rays were causing a whole bunch of damage to her body, and she actually died of ovarian cancer due to the radiation that she received from the X-rays. Her research was used by Watson and Crick to discover the structure of DNA. They did not actually recognize her in their findings of the DNA structure and until after she was dead. And unfortunately, she is not awarded the Nobel Prize because you cannot award it after you've passed away. You'll see that in your packet. And then I think actually we'll stop there for today. If you want to take a little bit more look at RNA and the structure of RNA, we'll get into that, but you'll see some of the basic comparisons as well. The rest of the time, you're going to start working on your WebQuest. All right.